into the left hypochondria. It has two surfaces, diaphragmatic surface and visceral surface. And from anatomical point of view, porta hepatis and two anterior posterior sulci divides inferior surface of the liver or visceral surface of the liver into four lobes: right lobe, left lobe, quadrate lobe, and caudate lobe. From histological point of view, the main digestive function of the liver is production and secretion of the bile, the complex substance required for the emulsification, hydrolysis, and uptake of fats in the duodenum. The liver is also the major interface between the digestive system and blood. Interface between digestive system and the blood. As the organ in which nutrients absorbed in the small intestine are processed before distribution throughout the body. If you remember, I told you portal vein receives blood from the intestine, large intestine and small intestine, and from the stomach and from the spleen, and send this blood into the liver. Yeah. About 70% of the blood entering into the liver is nutrient rich, because this blood came from
stomach,
large intestine and small intestine. Yeah?
yes.
After absorption of the nutrients from small intestine and some nutrients from large intestine, these nutrients go to the portal vein. And 75% of blood entering to the liver is nutrient rich and CO2 and O2 poor. Okay? Because it comes from another organ. It doesn't have more O2. Blood from the portal vein arising from the stomach, intestines, and spleen. And other 25% of the blood entering into the liver is from hepatic artery. And this blood has more O2, more oxygen. Is it clear?
Yes.
It has more oxygen. 75% of the blood entering into the liver has poor O2. And 25% of blood entering into the liver has
Oxygen.
more oxygen. Okay? Glison's capsule is a dense, irregular connective tissue. Glison's capsule, or capsule of the liver, is a dense, irregular connective tissue. Dense, irregular connective tissue. Invests the liver and send septa into the substance of the liver at the porta hepatis to subdivide the parenchyma of the liver into lobules. You see here, this is parenchyma of the liver. Here is outer serous layer, peritoneum, and this capsule is dense, irregular connective tissue. Dense, irregular connective tissue. This capsule is called Glisson's capsule. Glisson's capsule. Between parenchyma of the liver and Glisson's capsule, there is subcapsular space. Subcapsular space. The main and more abundant cells in the liver are called hepatocytes. Hepatocytes. Hepatocytes are key cells in the liver, are among the most functionally diverse cells of the body. They have many functions. If I ask you, which cell in your body has endocrine and exocrine function, you answer me, hepatocytes. Yeah? One cell has exocrine function and endocrine function. If I ask you, which organ has exocrine and endocrine functions? Pancreas. Yeah? Pancreas has exocrine part and endocrine part. But if I ask you, which cell has exocrine and endocrine functions? You answer me, hepatocytes. If I ask you, which organ has apocrine and merocrine secretions together? Apocrine and merocrine secretions together. Breast. The breast. Okay? Hepatocytes are cuboidal or columnar cells, large cuboidal or polyhedral epithelial cells with large, round, centrally located nuclei and eosinophilic cytoplasm. When the cytoplasm of the cell is eosinophilic, it shows this cell has abundant mitochondria. It has abundant mitochondria. This is large cuboidal cell or polyhedral cell with large centrally located nucleus, round nucleus, and eosinophilic cytoplasm, that this cytoplasm rich in mitochondria. The cells are frequently binucleated, and about 50% of them are polyploid. What's the meaning of polyploid? What's the meaning of polyploid?
A lot of color. We have a lot of colors.
Another colors?
A lot of color when we added some substance. It's called.
Have many shapes. Many shapes.
A chromosome. Have a lot of chromosomes. Have a lot of chromosomes.
In the normal condition, your cells are 2n chromosomes. They have 2n chromosomes. and each end is 23. Two N is 46, and N is 23. Yeah? Each set of chromosomes are 23. One chromosome number 1, number 2, number 3, each one has one. You know that in your cells, in the nuclei of your cells, there are 46 chromosomes. 44 or 22 pairs of these chromosomes are autosomal chromosomes. Autosomal. And one pair is sex chromosome. Do you know? Yeah? In the females, sex chromosomes are XX, and in the males, XY. Yeah? "23 chromosomes are a set, okay? And in normal condition, our cells have 2 N chromosomes. If instead of 23 multiple 2 or 46 or 2 N chromosomes, each cell has, for example, 3 N, 4 N. 23 multiple 3 multiple 4. This condition is called polyploid. Polyploid. Is it clear?
Yes.
Is it clear? The cells are frequently binucleated. They have 2 nucleus in one cell. Binucleated. Okay? 2 nuclei in one cell. With 2 to 8 times the normal chromosome number. Okay? There are more than 2 times of normal chromosome. Normal chromosome is 2N. Is it clear? Is it clear?
Yes.
You. What's the polyploid? 2 chromosome? 2N chromosome? No. 2N chromosome is normal.
Yes. More than 2 and yeah, more than 2.
More than 2N. More than 2N chromosomes. Is it clear?
Yes.
What's polyploid? You.
More than N chromosome.
No.
More than 2 chromosomes.
More than 2N chromosomes. Yeah. This condition is called polyploid. I told you, hepatocyte is a large cuboidal cell with 1 end or 2 surfaces. That these surfaces has many microvilli and lateral surfaces have attachment to neighboring hepatocytes, attached together. But on the lateral wall of the hepatocyte, you see here, you see a depression that when 2 hepatocytes attach together, make tight junction together. This area is called canaliculus. Intercellular canaliculus. I will tell about this canaliculus. Okay? This is the beginning part of the bile system. Secretion of the bile from hepatocyte drain into the canaliculus at first. Okay? I said hepatocytes have exocrine and endocrine functions together. Yeah? Exocrine and endocrine functions together. In addition to an exocrine function in the secretion of the bile components, hepatocytes and other liver cells process the contents of blood. Okay? Hepatocyte receives some nutrients from the blood and send something to the blood. Is it clear?
Yes.
Receives, for example, glucose from the blood and makes glycogen, store glycogen, and then glycogen becomes glucose and sent to the blood again. Okay? This is endocrine function. Release something into the blood. This is endocrine function. And bile production, bile secretion is exocrine function. Okay? Hepatocytes have many specific functions. Synthesis and endocrine secretion into the blood of the major plasma proteins. Major plasma proteins such as albumins, fibrinogen, apolipoproteins, transferrins. Okay? Some proteins in the plasma are synthesized in the liver, in the hepatocytes, and then released into the blood, such as albumins, fibrinogen, apolipoprotein, and transferrin and other proteins. Conversion of amino acids into glucose, gluconeogenesis. Conversion of the amino acid to the glucose. Occur in the hepatocytes. Okay? Gluconeogenesis. Amino acid deamination, producing urea, removed from blood in the kidneys. Detoxification or breakdown of conjugation and conjugation of ingested toxins. Ingested toxins, including many drugs, is another function of the hepatocyte. Detoxification of chemicals, drugs, alcohol. Okay? Storage of glucose in glycogen granules and triglycerides in small lipid droplets. Small lipid droplets. Storage of iron in complexes with the protein ferritin. Storage of iron. Okay? Liver parenchyma is organized as thousands of small hepatic lobules in which hepatocytes form hundreds of irregular planes irregular plates arranged radially around a small central vein. You see here, this is a classic hepatic lobule. It has one central vein and many hepatocyte plates, and between these hepatocyte plates there are many sinusoids. These are sinusoids. Okay? Hexagonal lobules of the liver are called classic lobules. Hepatocyte plates are supported by a delicate stroma of reticular fibers. Delicate reticular fibers. Peripherally, each lobule has 3 to 6 portal areas with more fibrous connective tissue, each of which contains 3 interlobular structures that comprises the portal teriad. Here is classic lobule, and on the corners of these classic lobules there is a portal area, and in each portal area there is portal teriad. Portal teriad is composed of a branch of hepatic artery, a branch of portal vein, and a branch of hepatic duct. Hepatic duct or bile duct. An arteriole branch of the hepatic artery that supplies O2, a venule branch of the portal vein with blood rich in nutrients but low in oxygen, 1 or 2 small bile ductules of cuboidal epithelium, branches of the bile conducting system. Is it clear?
Yes.
Between all of the anastomosing plates of hepatocytes of the hepatic lobule are important vascular sinusoids. There are sinusoids that emerge from the peripheral branches of the portal vein and hepatic artery and converge on the lobule central vein. This is in the center of the lobule. You see here, this is the classic lobule. This vein is central vein. These are hepatocyte plates, and between them there are many sinusoids. Around each lobule, this area is called portal area. Portal area. In this portal area there is connective tissue and a branch of the portal vein, a branch of the hepatic artery, and a branch of the bile ductus. Is it clear? These 3 organs are called teriad, portal teriad. Teriad means 3. Portal teriad. Here. Another name of portal area is called Kiernan space. Kiernan space. The venous and arterial blood mixes in these irregular hepatic sinusoids. The anastomosing sinusoids have a thin, discontinuous lining of fenestrated endothelial cells. Fenestrated endothelial cells surrounded by sparse basal lamina and reticular fibers. Okay? There are many features in the sinusoids. In the sinusoid, basal lamina is not continuous, sparse. Okay? And endothelial cells have many fenestra. This is fenestrated. And diameter of the sinusoid is not regular, it's irregular. Is it clear? Is it clear? Okay. Sinusoids are lined by the endothelial cells, similar to other capillaries. Okay? Are lined by the endothelial cells. The discontinuities and fenestrations allow plasma to fill a narrow perisinusoidal space. Perisinusoidal space or space of this. Perisinusoidal space or space of this. And directly, but the many irregular microvilli projecting from the hepatocytes into this space of this. This direct contact between hepatocytes and plasma facilitates most key hepatocyte functions that involve uptake and release of nutrients, proteins, and potential toxins. See here, this is a classical lobule. Classical lobules are hexagonal with portal areas or kernel spaces, teriots, at the periphery and the central vein in the center. Trabeculae or hepatocyte plates of the liver cells and stomos. Sinusoids are lined by sinusoidal lining cells, and sinusoidal lining cells are endothelium. Yeah? Are called endothelium. Similar to other capillaries. In addition to endothelial cells, there are specific macrophages that are called Kupfer cells. Kupfer cells. Specific macrophages of liver are called Kupfer cells. Within the space of these, fat accumulating cells may be noted. In the space of this, there are fat storage cells that are called E2 cells or Stranzelen cells. E2 or Stranzelen cells. Portal area housing bile ducts, lymph vessels, and branches of hepatic artery and the portal vein are surrounded by terminal plates composed of hepatocytes. You see here, this is a classic hepatic lobules, and on the corner of this hepatic classic lobules, there are portal area or kernel spaces. And in the portal area or kernel space, there is teriod, a branch of the bile ducts, a branch of the portal vein, and a branch of the hepatic artery. Yeah? These are teriods. In the portal area or kernel spaces. Bile passes peripherally within bile canaliculi. What is bile canaliculi? If you remember, I told you on the lateral wall of the hepatocyte, there are There are depressions that when two hepatocytes attach together and make tight junction around these depressions become bile canalicles without any lining, just is formed by the hepatocytes. Yeah?
Yes.
So are bile canalicles. Intercellular spaces between liver cells, hepatocytes, to enter bile ductules, then canals of Herring. This canaliculi enters to the bile canals of Herring. Bile canals of Herring. And cholangioles to be delivered to bile ducts at the portal areas. This is bile ductule, and these are canaliculums that enter to the Herring, canal of Herring, or bile canal of Herring, and goes to the bile ductules in the portal area. You see here, canal of Herring is lined by the cuboidal epithelium. That these cells are called cholangiocytes. Cholangiocytes. These cuboidal cells that line the canal of Herring are called cholangiocytes. Another classification of the lobules in the liver is called portal lobule. Portal lobule. If you remember, I told you classic lobules are hexagonal. Do you remember?
Yes.
With central vein in the center and portal area around this classic lobule. Do you remember?
Yes.
But in the portal lobule is a triangular area that portal area or Kiernan space is located in the center, and three central veins are located on the pieces. Okay? This is portal lobule. The pieces of triangular cross sections of the portal lobules are central veins. Thus, portal areas form the center of these lobules. The portal lobule is based on bile fluid. It's based on bile fluid. Okay? If I ask you what is the center, which part is the center of the portal lobule, you answer me portal area. Anterior. Portal anterior. If I ask you what's the center of the classic lobule of the liver? Central vein. Another classification of lobules is oscillus of Rappaport, or liver oscillus. Liver oscillus. Liver oscillus is a diamond shape. Liver oscillus is a diamond shaped part that long axis of this diamond is two central veins. This is two central veins. And short axis of this diamond is portal areas. Okay? From one portal area or Kiernan space to another one. The acinus of Rappaport in section is a diamond-shaped area whose line axis is the straight line between neighboring central veins, and whose short axis is the intersecting line between neighboring portal areas. The liver acidus is based on blood flow. This is based on blood flow. Yeah, this is based on blood flow. Okay, I told you classical lobules, portal lobules, and acidus of liver, or Rappaport, acidus of Rappaport. In addition to hepatocytes, there are two types of cells that functionally are very important in the liver. One of them is Kupffer cells. If you remember, I said sinusoids are lined by the endothelial cells. And between endothelial cells, among the endothelial cells, there are specific macrophages that are called Kupffer cells. These cells recognize and phagocytes aged erythrocytes, freeing heme and iron for reuse or storage in the ferritin complexes. Okay? After damaging the erythrocytes, heme molecules and iron released, and some cells, such as hepatocytes, absorb this. Kupffer cells are also antigen-presenting cells. And remove any bacteria or debris present in the portal blood. These are Kupffer cells. Kupffer cells are stellate cells. And they are modified macrophages. The origin of Kupffer cells are monocytes of the blood, similar to the macrophages. E2 cells, or Stranzelen cells, or fat storing cells, are located in the space of these. Where is the space of these? Between sinusoid and hepatocyte. There is a space that is called the space of this, or perisinusoidal space. Fat storing or E2 cells or Stranzelen cells are located in the space of these, the narrow space between the sinusoidal lining cells and the hepatocytes. E2 cells are believed to function in the accumulation and storage of vitamin A. Okay? This is cell for storage of vitamin A. But in the case of alcoholic cirrhosis, these cells also manufacture type 1 collagen. Okay? The cells synthesize collagen type 1, responsible for fibrosis of the liver. In the alcoholic cirrhosis, Stranzelen cells or E2 cells synthesize collagen type 1 and make fibrosis in the liver. Is it clear?
Yes.
You see here, this is a strong cell. E2 cells or hepatic stellate cells are believed to function in the accumulation and storage of vitamin A, I told you. These mesenchymal cells, which are difficult to see in routine preparations, also produce extracellular matrix components, becoming myofibroblasts after liver injury and cytokines that help regulate Kupffer cell activity. In the normal liver, most dense connective tissue is found only in the portal area, in each portal area, surrounding the blood vessels and bile ducts. In the liver cirrhosis, which occurs late in chronic liver disease, fibrosis, and proliferation of fibroblasts and hepatic stellate cells occur beyond the portal areas. The excessive connective tissue may disrupt the normal hepatic architecture and interfere with liver function. You see here, this is liver cirrhosis. Okay, fibers of the liver is called liver cirrhosis. The liver forms about 1 liter of bile per day, which is its exocrine secretion. Bile is delivered into the system of conduits. Bile canaliculi between hepatocytes, bile canaliculi, cholangioles, canals of Herring, interlobular bile ducts, which then directs into the bile into the common hepatic duct, and from there via the cystic duct into the gallbladder, storage organ associated with the liver. Is it clear? Okay. Bile is formed by the hepatocyte, and from hepatocytes drain into the canaliculi, intercellular canaliculi or bile canaliculi, and from canaliculi goes to the cholangioles and canal of Herrings and interlobular bile ducts and right and left hepatic ducts. Is it clear? And common hepatic duct drains to the cystic duct, and cystic duct goes to the gallbladder or storage of bile. Is it clear?
Yes.
The release of concentrated bile into the duodenum via the common bile duct is regulated by hormones of diffuse neuroendocrine system cells in the digestive tract. Bile is a green, somewhat viscous fluid composed of water, ions, cholesterol, phospholipids, bilirubin, glucuronide. Bilirubin, glucuronide. It's different from the bilirubin. Bilirubin, glucuronide, and bile acids. You see here, this is cystic duct, and here is common hepatic duct. Do you remember? Join together and make common bile duct. One of these components, bilirubin glucuronide, is a water-soluble conjugate of non-soluble bilirubin. It's very important that you know. Bilirubin glucuronide is water-soluble. Okay? Water-soluble. But bilirubin is not water-soluble, and it is toxic. Toxic breakdown product of hemoglobin. Bilirubin is toxic breakdown of the endorphin. When bilirubin becomes bilirubin glucuronide, it's water soluble and release, exit from urinary system. Okay? It's in the smooth endoplasmic reticulum of the hepatocytes that detoxification of bilirubin occurs. If I ask you, detoxification of bilirubin occur in which organelle of the hepatocyte? You answer me, smooth endoplasmic reticulum. Smooth endoplasmic reticulum. If you remember, the smooth endoplasmic reticulum has many specific functions. Do you remember? Synthesis of phospholipids in some cells. Synthesis of phospholipids. Detoxification in the hepatocytes. And calcium storage in the muscle.
Yes.
That the name of smooth endoplasmic reticulum in the muscle is sarcoplasmic reticulum. Sarcoplasmic reticulum. Do you remember? Yeah? One of the specific functions of the smooth endoplasmic reticulum is detoxification. And detoxification of bilirubin occurs in the SCR of the hepatocytes. Is it clear?
Yes.
Bile flow progresses in a direction opposite to that of the blood, that is from the center of the lobule to its periphery. Bile canaliculi are the smallest branches of the biliary tree or bile conducting system. They empty into bile canals of herring composed of cuboid epithelial cells called cholangiocytes. Yeah? I told this one. The short bile canals quickly merge in the portal areas with the bile ductules lined by cuboidal or columnar cholangiocytes and with a distinct connective tissue sheath. Bile ductules gradually merge, enlarge, and form right and left hepatic ducts, leaving the liver. Two right and left hepatic ducts combine to form the common hepatic duct, which runs near the liver with the hepatic artery proper and portal vein in the free margin of the lesser omentum. Yeah? The cystic duct from the gallbladder, joins to the common hepatic duct. This completes the formation of the common bile duct, or common bile duct. At this point, the common bile duct lies to the right of the hepatic artery proper, and usually to the right of and anterior to the portal vein in the free margin of the lesser omentum. The omental foramen is posterior to these structures at this point. Omental foramen, another name of omental foramen is Winslow's foramen. Winslow's foramen. Which structure is superior border of the Winslow's foramen? Caudate process. Caudate process. Which structure is the inferior border of the Winslow's foramen? First part of the duodenum. Which structure is the posterior border of the Winslow's foramen or omental foramen? Inferior vena cava. And anterior border of the Winslow's foramen is formed by the free border of the lesser omentum. And in the free border of the lesser omentum, there are three structures: common bile duct, hepatic proper artery, and portal vein. Do you remember? The common bile duct continues to descend passing posterior to the superior part of the duodenum, first part of the duodenum, before joining with the pancreatic duct, or main pancreatic duct, or Wilson duct, to enter the descending part of the duodenum at the papilla duodeni major, major duodenal papilla. Hepatic, cystic, and common bile ducts are lined with a mucous membrane having a simple columnar epithelium of cholangiocytes. Simple columnar epithelium. Hepatic, cystic, and common bile ducts. Okay? Common bile duct, cystic duct, and common hepatic duct are lined by the simple columnar epithelium of cholangiocytes. Lamina propria and submucosa are relatively thin, with mucous glands in some areas of the cystic duct and surrounded by a thin muscularis. This muscle layer becomes thicker near the duodenum, and finally in the duodenal papilla forms a sphincter that regulates bile flow into the small intestine. This sphincter is called Oddi's sphincter, sphincter of Oddi. I told you, joining the common bile duct to the main pancreatic duct in the ampulla of water, and around the ampulla of water there is sphincter of Oddi. Gallbladder has 3 layers: tunica mucosa, tunica muscularis, and tunica serosa. The mucosa has numerous folds that are particularly evident when the gallbladder is empty. There are many folding in the mucosa. This is mucosa, this is muscularis layer, and here is serosa. Epithelium of gallbladder is a simple columnar epithelium. Simple columnar epithelium. You see here, simple columnar epithelium. Lamina propria is drawn into intricate folds that appeared in the distended gallbladder. You see here, there are many folds. Rokitansky-Aschoff cyanosis, epithelial diverticula may be present here. Some diverticula, epithelial diverticula, that are called Rokitansky-Aschoff cyanosis, are seen in some area. Muscularis externa is composed of an oblique-oriented smooth muscle layer. Here is muscular layer, and serosa attached to the inferior and lateral surfaces of the gallbladder, and superior surface of the gallbladder is lined by the adventitia because it doesn't have peritoneum attached to the inferior surface of the liver. Okay. After 15 minutes rest, come back. Okay. Pancreas is a retroperitoneal organ that is located posterior to the stomach. It extends across the posterior abdominal wall from the duodenum on the right to the spleen on the left. The pancreas is retroperitoneal except for a small part of its tail that is located in the
inferior.
lienorenal ligament. Lienorenal ligament. Pancreas has 20 centimeter length. 20 centimeter length, and it has head, uncinate process, neck, body, and tail. Okay? The head of the pancreas lies within C-shaped concavity of the duodenum. Here is duodenum, and here is head of the pancreas. Projecting from the lower part of the head, is the uncinate process. This is uncinate process. In the inferior part of the head, there is uncinate process, which passes posterior to the superior mesenteric vessels. You see here, this is superior mesenteric artery, and this is superior mesenteric vein. Here is posterior to the superior mesenteric vessels. This is posterior view. Yeah, posterior view of the duodenum and posterior view of the head of the pancreas. Here is uncinate process, here is head, and here is neck. The neck of pancreas is anterior to the superior mesenteric vessels. Anterior to the superior mesenteric vessels. And posterior to the neck of the pancreas, the superior mesenteric and splenic veins join together and form portal vein. Okay? Posterior to the neck of the pancreas, superior mesenteric vein and splenic vein join together and makes portal vein. You see here, this is portal vein. This is superior mesenteric vein, and here is splenic vein that makes together and join together and make portal vein. The body of pancreas is elongate and extends from neck to the tail of the pancreas. The tail of the pancreas here passes between layers of splenorenal or lienorenal ligament. Here is pancreatic duct, begins in the tail of the pancreas. Pancreatic duct begins in the tail of the pancreas. It passes to the right through the body of the pancreas, and after entering the head of the pancreas, turns inferiorly. This is main pancreatic duct or Wilson duct. In the lower part of the head, the pancreas, or head of the pancreas, the pancreatic duct joins to the common bile duct here, that this area is called ampulla of water or hepatopancreatic ampulla. The joining of common bile duct and pancreatic duct forms the hepatopancreatic ampulla or ampulla of water, which enters to descending or second part of the duodenum at the papilla duodeni major. Surrounding the ampulla is the sphincter of ampulla or sphincter of Oddi. Sphincter of Oddi, which is a collection of smooth muscles. Accessory pancreatic duct or Santorini's duct of Santorini, empties into the duodenum just above the major duodenal papilla, superior to the duodenal papilla duodeni major, in the papilla duodeni minor or minor duodenal papilla. The arterial supply to the pancreas includes the gastroduodenal artery. If you remember, I told you there are two superior pancreatico-duodenal arteries, anterior and posterior superior pancreatico-duodenal arteries, from anterior surface and posterior surface of the head descend. And from superior mesenteric artery, inferior pancreaticoduodenal arteries, anterior and posterior arteries, ascend and join on main anastomosis with the superior, anterior and posterior pancreaticoduodenal arteries. This is joining of anterior, superior, and anterior, inferior pancreaticoduodenal arteries. Superior pancreaticoduodenal arteries are related to the gastroduodenal artery, branches of the gastroduodenal. And inferior pancreaticoduodenal arteries are related to the superior mesenteric, branches of the superior mesenteric. Posterior superior pancreaticoduodenal artery, posterior superior pancreaticoduodenal artery forms the gastroduodenal from the gastroduodenal artery, and posterior inferior pancreaticoduodenal artery from the inferior pancreaticoduodenal artery is the branch of the superior mesenteric. I told you about posterior and anterior branches. Greater pancreatic artery from the inferior pancreatic artery, a branch of the splenic artery, and dorsal pancreatic artery from the inferior pancreatic artery, a branch of the splenic artery. You see here, these arteries supply body or trunk of the pancreas. Pancreas is an exocrine and endocrine organ together. I told you, if I ask you which cell has exocrine and endocrine functions together, the answer be hepatocytes. Which organ has exocrine and endocrine functions together? Pancreas. Pancreas produces both digestive enzymes and hormones. Pancreas has a thin capsule of connective tissue from which septa expand to cover the larger vessels and ducts and to separate their parenchyma into lobes. This is pancreas, and it has exocrine, pancreatic acini, and endocrine part that are called Langerhans islands. Okay? Secretion of beta cells, alpha cells, glucagon, insulin, PP. Yeah? These are from endocrine part of the pancreas. And there are exocrine enzymes that release into the duodenum. For example, elastase. Okay? Secretory acini are surrounded by a basal lamina that is supported only by a delicate sheet of reticular fibers with a rich capillary network. Endocrine function of the pancreas involves primarily smaller cells similar to enteroendocrine cells located in the variously sized clusters called the pancreatic islets or islets of Langerhans. This is islet of Langerhans, and these are exocrine acids. Every day, the exocrine pancreas produces approximately 1.5 liter of an alkaline fluid rich in digestive enzymes and proenzymes, which is delivered to the duodenum via the pancreatic duct. This is exocrine part. Each pancreatic acinus consists of several serous cells several serous cells surrounding a very small lumen without myoepithelial cells. Without myoepithelial cells. Okay? If I ask you, what's the difference between exocrine acini or serous acini of the pancreas and serous acini of the parotid gland? You answer me?
Myoepithelial cells.
Lack of myoepithelial cells in the pancreas. It's one of the differences. There is another difference I will tell. Okay, one of the differences is acini, serous acini of pancreas don't have myoepithelial cells. Don't have myoepithelial cells. Acinal cells are polarized with round basal nuclei and numerous zymogen granules typically of protein secreting cells. This is protein secreting cells, similar to serous cells in the parotid gland, submandibular gland, I told you. Yeah? Each acinus is drained by a short intercalated duct of simple squamous or low-cuboidal epithelium. The initial cells of this small duct extend into the lumen of the acinus as small pale staining centroacinar cells that are unique to the pancreas. Okay? If I ask you, what is difference between parotid gland serous secretions or serous parts, acids, and serous acini of pancreas, you answer me, there are two differences.
Myoepithelial cells.
In the pancreas, serous acini don't have myoepithelial cells. And in the pancreas, inside of the serous acini, there are centroacinar cells. Centroacinar cells are continuation of squamous or flat cells of the intercalated duct that enter into the acinus. Is it clear?
Yes.
I will ask this. I will ask. You cannot answer me. I don't know why. Okay? Okay? There are two differences between serous acini of the parotid gland and serous acini of the pancreas. Okay? Lack of myoepithelial cells and centroacinar cells in the acini of the pancreas. Is it clear?
Yes.
Cells of the intercalated duct secrete a large volume of fluid rich in bicarbonate ions, which alkalinize and transports hydrolytic enzymes produced in the acid. The intercalated duct merge with intralubular ducts and larger interlobular ducts which have increasingly columnar epithelia before joining the main pancreatic duct that runs the length of the gland. Okay? This intercalated ducts with cuboidal epithelium join together and make intralobular, and intralobular ducts join together, make interlobular ducts. Interlobular ducts have simple columnar epithelium. And interlobular ducts join together, make main pancreatic duct. Is it clear? Exocrine secretion in the pancreas is regulated mainly through two polypeptide hormones produced by enteroendocrine cells or diffused or endocrine cells of the small intestine. Cholecystokinin stimulates enzyme secretion by the acinar cells, and secretin promotes water and bicarbonate ion secretion by the duct cells. Okay? Cholecystokinin and secretin. In acute pancreatitis, the proenzymes may be activated. Proenzymes may be activated and digest pancreatic tissue, leading to very serious complications. Possible causes include the infection, gallstone, alcoholism, drugs, and trauma. Okay, digestion of the pancreas by proenzymes of the activation of proenzymes of the pancreas. Chronic pancreatitis can produce progressive fibrosis and loss of pancreatic function. Pancreatic cancer, which is usually a carcinoma of duct cells, can arise anywhere in the gland, but occur often in the head of the pancreas, near to the duodenum. The tumor is usually asymptomatic until it grows and metastases are well advanced, leading to the low rate of early detection and subsequent high rate of mortality. Metastasis may be facilitated by the relatively sparse connective tissue around the ducts and musculature of the pancreas. Do you have any question about pancreas? Okay. You will read, you will study Langerhans islands in the endocrine system. Okay? Alpha cells, beta cells, B cells. You will study in the endocrine system. Spleen is a part of the lymphatic system, but it has a near relationship with the digestive system. Location is in the left hypochondrium, and it has two surfaces. I told you, it has two surfaces. Parietal surface or costal surface or diaphragmatic surface is convex, and visceral surface is concave. And visceral surface has a linear hilum and three depressions for relationship with the stomach, with the left colic flexure, and with the left kidney. Yeah? The spleen is surrounded by visceral peritoneum except in the area of the hilum on the medial surface of the spleen. Splenic hilum is the entry point for the splenic vessels, and occasionally the tail of the pancreas reaches this area. This is hilum. Spleen is connected to the greater curvature of the stomach by the gastrosplenic or gastrorenal ligament, which contains the short gastric and gastroomental vessels. This is gastrorenal gastrosplenic ligament. And connects to the left kidney by the splenico-renal or lieno-renal ligament, which contains splenic vessels and tail of the pancreas. Diaphragmatic surface or costal surface of the spleen is convex and is covered by ribs number 9, 10, and 11. 9, 10, and 11. Visceral surface of the spleen has relation to stomach, left conic flexure, and left kidney. Do you have any questions?